Welcome to today's lesson on using scale and magnitude to make calculations in biology. In this session, we will focus on understanding orders of magnitude and standard form. This skill is essential for accurately representing and working with the incredibly small or large quantities that we often encounter in cell biology. Today, we will explore the concepts of scale and magnitude, as well as teaching you how to calculate and express numbers using orders of magnitude and standard form. This lesson will involve practical exercises to ensure you can confidently apply these techniques. So far, you have learned that the cell is the basic unit of life and that it contains smaller subcellular structures called organelles. These structures are incredibly small and require orders of magnitude to understand their relative size. Building on this foundation, learning about scale and magnitude will enhance your ability to quantify and compare biological structures and processes. This concept advances our biological understanding by enabling us to make precise calculations and representations, which is crucial for research, diagnostics, and developing medical treatments. Let's get started. First, it is essential to understand that cells are extremely small. We can use scale to compare the relative sizes of these very small structures. When we talk about scale, we mean the relative size of an object compared to another. For example, the diameter of a typical red blood cell is approximately 6 micrometers in diameter, while a cheek cell is roughly 60 micrometers in diameter. To compare the sizes of these two cells, you can divide the size of the cheek cell by the size of the red blood cell. Hence, 60 micrometers divided by 6 micrometers is equal to 10, meaning a cheek cell is 10 times larger than a red blood cell. Orders of magnitude provide us with a useful way to understand the relative size of cells when compared to each other. An order of magnitude describes the scale or size of a value by comparing it to a fixed reference point. Typically, when working with orders of magnitude, we use factors of 10. For example, if you increase a number by one order of magnitude, you are multiplying it by 10. If you decrease a number by one order of magnitude, you're dividing the number by 10, which is equivalent to multiplying by 0.1. Let's take a look at some common orders of magnitude. 100 is 2 orders of magnitude greater than 10, because 100 equals 10 to the power of 2. 1000 is 3 orders of magnitude greater than 10, because 1000 equals 10 to the power of 3. 0.001 is 3 orders of magnitude smaller than 10, because 0.001 equals 10 to the power of minus 3. In cell biology, orders of magnitude help us to compare the sizes of various cells and subcellular structures in a clear and concise manner, and is used as part of the standard form notation. First, it is essential to understand that cells are extremely small. We can use scale to compare the relative sizes of these very small structures. When we talk about scale, we mean the relative size of an object compared to another. For example, the diameter of a typical red blood cell is approximately 6 micrometers, while a cheek cell is roughly 60 micrometers in diameter. To compare the sizes of these two cells, you can divide the size of the cheek cell by the size of the red blood cell. Hence, 60 micrometers divided by 6 micrometers is equal to 10. This means that a cheek cell is 10 times larger than a red blood cell. Now, let's explore standard form, which is a method to express numbers using orders of magnitude. Standard form is written in the format a multiplied by 10 to the power of n. Here, a is a number between 1 and 9, and n is a whole number. Standard form notation makes it easier to write and work with very large or very small numbers. For example, 2000 in standard form is written as 2 multiplied by 10 to the power of 3. Similarly, 0.01 when written in standard form is equal to 1 multiplied by 10 to the power of negative 2. To write a number in standard form, you need to divide the original number until it becomes a number between 1 and 9. Then, you need to express this value as being multiplied by an order of magnitude. For example, 200 divided by 100 is equal to 2, as 100 is the equivalent of 10 squared. 200 can be written in standard form as 2 multiplied by 10 to the power of 2. Let's look at another example. To write the number 0.00035 in standard form, we need to first turn it into a number between 1 and 9. To do this, we can multiply 0.0035 by 1000 to give us 3.5. This is the equivalent of dividing 0.00035 by 0.0001. Therefore, as 0.0001 is the equivalent of 10 to the power of minus 4, 0.00035 can be written as 3.5 multiplied by 10 to the power of minus 4. Let's go through some more practical examples to reinforce these concepts. In our first example, let's compare cell sizes. The diameter of an E. coli bacterium is approximately 2 micrometers, and the diameter of a mitochondrion within a cell is roughly 0.5 micrometers. To compare these, we need to divide the size of the bacterium by the size of the mitochondrion. 2 micrometers divided by 0.5 micrometers is equal to 4. This means that the E. coli bacterium is 4 times larger than a mitochondrion. Next, let's complete an example question on orders of magnitude. Say we want to find how many orders of magnitude a 20-meter tree is larger than, when compared to a 0.00002 meter cell. To achieve this, we need to divide 20 by 0.00002, which equals 1 million. 1 million can also be written as 10 to the power of 6. It can therefore be stated that the tree is 6 orders of magnitude larger than the cell. Finally, let's look at an example of expressing sizes in standard form. Suppose we want to express the width of a leaf palisade cell in standard form. The width of a leaf cell is 0.027 millimeters. To express this in standard form, we must first convert 0.027 into a number that lies between 1 and 9. To turn 0.027 into a number between 1 and 9, we need to multiply it by 100, which gives us 2.7. This is the same as dividing 0.027 by 0.01. As 0.01 is equal to 10 to the power of minus 2. In our final answer, we have 0.027 is equal to 2.7 multiplied by 10 to the power of minus 2. In today's lesson, we delved into the concepts of scale and magnitude to help you understand how to make calculations in cell biology using orders of magnitude and standard form. First, we explored the concept of scale, helping you to grasp how vastly different sizes can be effectively compared and calculated. We learned that we can calculate scale by dividing one measurement by another. This involved understanding orders of magnitude, a method that allows how to express differences in size through powers of 10. We also focused on standard form, which enables the representation of very large or very small numbers, making them easier to work with in calculations and scientific communication. To express a number in standard form, we divide it so that it becomes a number that lies between 1 and 9. Then, we must express our value as that number multiplied by an order of magnitude. Understanding scale and magnitude enhances your capability to work with the very small and large numbers that are frequently encountered in fields of biology. Watch the rest of the GCSE biology course at Sophos Education.